Our next speaker is Victoria Hermanson. Victoria came to Scripps as an environmental manager from the U.S. Corps of Engineers in Sacramento, where she spent time doing CEQA analysis and other kinds of environmental analysis. She came to Scripps because she was interested in marine policy and wanted to get even more of the scientific foundation under her feet. She loved birds, and the surveys that she did with the U.S. Corps of Engineers was one of her favorite parts about her previous job. So it's no coincidence that she happens to be doing her capstone with seabirds today. So the title of Victoria's presentation is Through the Eyes of a Penguin, Estimated Krill Densities Using Animal-Borne Video Loggers.
Thanks, Samantha. So I spent my time working on my capstone with an amazing group of people with the Antarctic Ecosystem Research Division within the National Oceanic and Atmospheric Administration. I used visual data that was collected from the back of a penguin in a new framework or software called VIAMI in order to estimate densities of krill in the Antarctic. But before I dive into the details of my project, I sought the help of our resident penguin expert and created a video to give a little background on what this division does.
Our role here at the Antarctic Ecosystem Research Division is manyfold, I suppose, but one of the main things that we do is to advise policymakers in the Southern Ocean about fishing. And specifically, most of our work here focuses on the krill fishery. It's at the crux of an issue about how do fisheries and how do climate change impact predators. And so it starts with their impacts on the target species. So in this case, in the Antarctic, there is a fishery for krill. And it's a big fishery, it's poised to grow. And there's a lot of concern that as it grows, it could impact predators in that ecosystem, penguins being one of those. But if krill went away, I think there are a lot of animals that we consider to be dependent on krill, and they would certainly suffer. Undoubtedly, there's probably some fishes and other things like that take advantage of this enormous biomass of krill and focus predominantly on that. The question is whether or not, as those populations become depleted, whether those animals are capable of eating anything else. There's not another comparable resource to support predators in the volumes that exist now in the absence of krill. Just recently, we started using a new camera, a new technology for us, and so we've been starting to collect video footage of penguins actually eating krill in the ocean, which is the first time that we've been able to observe that directly. So the benefit of using the video loggers on penguins is that it takes you right to that interaction. It takes you beyond having to make inference about what those animals are doing, because now we can actually observe how that animal's behaving around its prey. And up until video cameras were actually there, unless you were underwater watching them, you really had to make some kind of inference from the way that they were moving underwater, or whether it had really encountered prey when it was making those movements. Here we can see that, no, it's encountering that prey, it just ate that prey, and hopefully through that then we'll be able to understand more about the distribution of animals in the ocean and how those distributions vary in space and time, and then how predators are impacted. Our ultimate goal with all of the data that we collect is to hopefully move the management away from sort of simplistic, old school approaches to managing the krill fishery into a more modern fisheries management era that takes ecosystem components into account and actually kind of has a feedback mechanism so that the catch limits and spatial allocations might be adjusted based on information from the actual predators that are being impacted by the fishery. So that's something that this group has been working on for a long time. It's trying to bring that monitoring and from our field sites and from our research surveys into that full-fledged ecosystem approach to management in the Southern Ocean.
Okay, so now I want all of you to look at one of your thumbs. That is roughly the length of an average adult krill, so they are tiny creatures. The Antarctic krill is said to have the highest biomass out of any animal in the world, so it's very hard to study these animals because of their location, size, and behavior, especially when it comes to putting a value on a three-dimensional density of them. NOAA has been using various methods to monitor krill, such as ship-based surveys, moorings, and autonomous gliders, but none have been used on penguins to get that first-hand look at the direct predator-prey interaction between them and krill. I helped NOAA by creating algorithmic models to estimate how dense krill populations can get by using the data collected from the cameras that Jefferson was talking about in the video. This is the kind of footage that I was working with. Before I get into the details, I want to give you a better idea of where these penguins are foraging. This is the Antarctic Peninsula. Within the Antarctic Peninsula, my study area is shown in the shaded green region. And within that shaded green region, this is an even closer look at the study area, and it's showing us the foraging routes of 2 chinstrap penguin individuals. It's a good representation of the area that my chinstrap penguin is covering on its journey. So who cares, and why is using technology like this so important? Krill are a keystone species, which means that most animals that inhabit the Southern Ocean rely on krill both directly and indirectly. Specifically for penguins, it is their primary food source. So because of these cameras, instead of making assumptions about what these animals are actually encountering or seeing in their field of view, we have the ability to experience it with our own eyes too now, allowing us to see the dense populations of krill through the eyes of a penguin. So I just wanted to give you all another reminder of what the footage looked like. This is one of the more exciting clips. The majority of the video is open water, travel, and surfacing events. So as you can see, it's extremely fast and hard to tell just how much krill the penguin is encountering. Also, this is only seconds out of an entire 5-hour foraging trip. There are multiple penguins that NOAA has fitted with these loggers, which means there's a lot of data to go through. That's why they're taking advantage of more advanced technology, and that's where my use of a new framework to analyze this data came into play. This framework is what I was applying to the visual data to create algorithmic models. Video and Image Analytics for a Marine Environment, or VIAMI, is an open source framework that is intended to be used for underwater video imagery analytics. It was just released last summer, and it's only been used by a handful of people. For this most recent field season, NOAA collected 4 different foraging videos from 2 Gen 2 penguins and 2 chinstrap penguins. The total number of frames for just one chinstrap video I was working with came out to 270,000 frames within the 5-hour event. So the idea was, instead of manually going through each of those frames, after creating models through VIAMI, there would be a more streamlined process to find krill that's being encountered. So let's go through the process. There were 4 main steps. First, I had to index the frames to create a massive database. Then I had to train models to find specific images throughout the video. For my final product, I ended up creating 4 different models. In order to do that, I had to select an example image to be used as a basis for creating the model. This is an example of one of the images I used to create as a model for finding closer images of krill. Once I selected this image in the green box shown, the software would use that to try to find similar images throughout the video. This meant going through an iterative process of selecting correct and incorrect images, shown in the green and red, as the software gave me results. Once I went through classifying each frame, I would hit refine, and it would give me another set of images to classify based on the feedback that I gave it in order to make the model more accurate the next time around. After 4 or 5 refinements, the model could be exported to be used on other video imagery. Then, each of the 4 models that were created were processed on the 5-hour video to store each of the frames into a specific class. I went through so many trials of creating different models and figuring out the best settings to use, but I came to a point where these were the final example images I used as a basis for creating each of the models, which then resulted in what I call these final 4 classes: all krill, close krill, penguin friends, and penguin surfacing. All 270,000 frames were sorted into one of these classes based on what the models were trained to detect for each. As much as I would love to continue looking at penguin butts, I'm only going to focus on the krill classes for the purposes of this study. These are examples of the results from the detected images that were classified into the all krill class. The idea was to try to get higher krill counts and more swarm activity. And here are some of the results of the detections that came from the close krill class. But now we have to come back to the main goal of this study. How did I end up from there to calculating density? It required two main steps. I took the count of krill from a frame and divided it by the standardized volume in the penguin's field of view in the shaded region shown. So in layman's terms, this means it gives me how many krill are in X meters cubed of water. That number gave me an estimate of the amount of krill in the penguin's field of view for any given frame. So what did we find out? The close krill class had average counts and densities that were lower than the all krill class. This makes sense because the models were trained to detect a closer and in turn lower amount of krill in each frame. All krill is supposed to encompass more swarm-like behavior. After looking at all the frames that were sorted, it turned out that the models were pretty effective in detecting the correct images and sorting them into the right classes. Classifier model results were 100% correct for close krill and 97% correct for all krill. Another important note is that both krill classes combined made up under 1% of the total 270,000 frames. This tells us that penguins aren't encountering krill too often in a single foraging trip, which means having a framework like Beyond is so much more effective when it comes to processing hours upon hours of visual data that NOAA is acquiring. So I learned that creating these models showed to be very effective. Being able to utilize these models for NOAA's future studies will significantly cut down the time spent on data analysis of underwater imagery, which is becoming harder to keep up with as the years go by. Using this data to have a more comprehensive understanding of the way krill are behaving during predator-prey interactions can provide useful information in managing the krill fishery and hopefully influencing marine policy. This was the first time krill densities have been estimated by using the video loggers on penguins. Ideally, NOAA will be able to use the methodology I implemented for their future studies for a more efficient video imagery analysis process. Thank you. Crystal?
First of all, it's brilliant. Second, thank you. How did those video loggers actually stay on the penguins, and then how is the data collected afterwards?
I'm not sure exactly how, but from what Jefferson told me, it's a process of using glue in addition to zip ties. And they don't stay on the penguins, however, sometimes they're predated by other things, so eaten or they fall off. But it's just like they get it from satellites, I believe. Yeah. Any other questions?
Does the video logger also track position of the penguins so that you can map where the swarms are?
It doesn't, but they do have other trackers that they use in addition to the video loggers to track that.